There you go. Hello, everybody. Is it okay? So first of all, I'd like to thank Fernanda and Raf for organizing this very nice school and for inviting me to give the course. Thank you for being here after the whole day. I hope your brains are still there. I will talk a lot about our brains. I ask you to really try to follow me and ask if you want me to speak slow or repeat. So the idea is for us to learn something. Any of you working with neuroscience, neurons? Okay, great. One, just. And Pedro, my student? Two. Please. So I think in the course for very beginners, for people that are not from the area, but I mean, I'm a physicist working in neuroscience, so it's a mix, it's interdisciplinary, perhaps many different parts, different people will understand, will think it's easy, and other parts will be difficult for other ones. So really, let's try to go together, okay? So first thing, is the brain a machine? In machine learning area, is the brain a machine that interprets reality? So can you look to this picture and tell me, don't tell me, but think, can you realize what's changing every time it blanks? When you realize, we will never be the same again. So if you tell loud, everybody will realize, so let's just wait a little bit. Who is not seen yet? Someone? No problem. It's the common thing is that you are not seen yet. And after I tell you, it's going to lose the magic. Or sometimes you just realize. Would you like to tell me what is changing? Now you can tell. The bar. So the biggest thing in the screen probably is changing, but yes, it's nice, no? But sorry, we can never unsee it. So my PDF doesn't work, so it's not blanking anymore, but this is the idea of reality. So the external stimuli is the same since the beginning of my question, but you don't realize what's going on, and then in a certain moment, you realize, and then you cannot, don't see it anymore. So the external stimuli is the same, but you are interpreting in a different way. So you are interpreting the reality or the stimuli that arrive in your eyes in a different way. So what's going on in our brain when we realize that or when we do not realize that? So this is the kind of question I'm interested in my scientific life. And this is the way I use biological neural networks to do science. I will try to show you that depending on the way you use or you propose your questions, your scientific questions, you will be good for you to use biological neural networks or use artificial neural networks. And I will try to go together with a very brief introduction to biological neural networks, but doing the parallel with very simple neural networks presented by Ralph, and trying to say in which moments we can put more biological ingredients and in which time we should use one or another. But I'm usually thinking like a neuroscientist. I mean, I like to understand the brain. And we could use like computer idea, medical ideas, linguistics can ask the same kind of questions, how the brain works, or physicists, mathematicians. So different people, different questions, different way to approach the problem. So here, during the talk, in my mind at least, will be all the time two things together. So experimental data from the brain, or cognition, like we can do a task, we can realize that the bar is moving. And if we measure the brain activity, how our neurons are communicating, we will have a signal. So we could measure this signal in a fMRI machine. We could measure it with electroencephalogram. We could measure in some very specific cases inside the brain, like people that need to go to surgery, epileptic patients, that you have to put a needle inside the brain, and then you have the electrical activity of very specific areas. Then you can see what's going on in this biological neural network as a signal, as a dynamical process. And we, as a physicist or a computational neuroscientist, we would like to propose a model. So we'd like to use biological neural networks math computer to develop something that looks similar to what's going on in a real brain. So I think that's my main point today. So we can have experiments in vitro. We can measure real neurons that have just draw in the beginning of this class, neural networks. It starts with a neuron, but then the neuron more or less disappeared. So I'm going after that. I'm really looking to neurons, to electrical activity, and ions, and a little bit of biology, so you can help me with that also. But the idea is that we have experiments in the brain, in cognitive sense, and we have experiments in slices, in small tissues. And we can have this time series, and you can think about models to reproduce these signals. So we can try to develop models that reproduce these signals, or in a following step, that reproduce cognitive tasks. So if we think about computational neuroscience, we can think that we are interested in looking for electrical and chemical signals of the cells. We would like to understand how do we respond to this external stimuli of the world. How do we interpret reality? Things that are arriving to our senses, like through the eyes, the ears, and we are interpreting and acting on it. So this would be the how the brain works, okay? But in the machine learning area, we can ask, can we build a machine that interprets reality? Can we build a brain? So what can we do? If we understand, can we build? Can we build even if we don't understand? So we can play with this kind of question, but this is what is following my my surrounding of ideas. So can we build a machine that interprets reality, so that can reproduce, for example, this cognitive task of recognize the bar after a while? So what would be necessary to build this machine? Actually, I built 2 of that, Enrique and Kyle. They are my sons, and they are very good to interpret reality and to classify dogs. So far, the best biological neural network that I have implemented. I tried a lot with my computer, but they are very better. So, except from the fact that we can reproduce brains, like we women, can I do that with my computer? So can I use math and computers to build a machine that is similar to the brain? So that's what we are trying to see what can we do. So from one point of view, we will have these artificial networks, perceptrons, deep learning neuronal networks, spiking neuronal networks, or we can have biological neural networks, and there is no clear boundary between one thing and the other, so you can put ingredients of biological stuffs, you can put ingredients of artificial networks. So I will try to make the point that we could think about like this plane here of different cognitive tasks and how would be our neural networks. So real brains could classify dogs, interpret reality, do a lot of things, do not other things, or it's very hard to do and computers can do. And depending on the task, it's enough a very simple artificial neural network, or you have to go more complicated biological neural networks. So this is what we are going to come again many times during the presentation. So what's the simplest model to reproduce a task? So what would be the simplest perceptron that could classify digits, or dogs and cats? And what model is more efficient? So what kind of ingredient you can put to learning fast, or learning rates, or how can you go there? This is a kind of question. You can come here and try different neuronal networks. So if we are more like, what are the important biological ingredients? So when you have some kind of disease, you cannot recognize the bar anymore. So I'd like to say if it's sodium or potassium that is changing your brain, if you are not plastic anymore, if neurons are dying. So if you like to go for more medical questions, you need to understand, even if you do not understand the brain, you would like to understand how the biological neural network is actually doing some task. So we can sometimes not understand the brain, but we know that when the myelin is gone, you have problems with memory. So we start to improve our medical solutions, considering that you understand better few biological neural networks. So for the most of the time, I will be here thinking about these biological ingredients and how the brain is performing the task. And what kind of ingredients I need to be here. So as a first example, I would use the binding problem, which is close to a classify question. The binding problem that you see that and you say, oh, this is an apple. So what does it mean? It's a classification problem. When you see an apple, you recognize it's an apple. So we can think in terms of machine learning, in neuronal networks. Could we develop a machine that do that? Yes, we give a lot of pictures of apple, and then after a while, we have this. But when we recognize an apple, what's going on? So we are not just classifying. We are remember someone that divide an apple with us at some point. We know that it's food. We remember a banana because typically apples and bananas are together. We remember balloons because of the shape. So in neuroscience, this binding problem is not solved in the sense that we don't really understand how all the details go, but there is a hypothesis and many evidence that the binding problem, at least, is related to neuronal assemblies, to neurons that fire together, and to neurons that are responsible for different features. So if you want to understand how the brain performs the task, we can measure the brain when we are recognizing apples, when we are classifying apples, and see that many neurons fire together. There is a famous experiment in neuroscience that measure people when they see the picture of Jennifer Aniston. And they find for many different people that there is a neuron that fires when you see the Jennifer Aniston, so they start to call the neuron of Jennifer Aniston. So, never heard about that? Okay, great. That's and then they change for the grandmother neuron name, because perhaps you have a neuron that fires when you remember your grandmother. But it's not that you have one neuron, but when we pitch, when we record few neurons in the brain, in a lot of neurons, you will probably find just one that is part of a group that fires together when you recognize Jennifer Aniston. And this neuron will probably fire for other things because you do not have a neuron that is just for Jennifer Aniston, I hope. But you probably not show the correct thing that makes that neuron fires again when see, I don't know, another blonde actress. So this is the binding idea. This is the kind of question. Can we develop a biological neuronal network that not just classify apples or Jennifer Anistons, but also do something that is similar? So make assemblies that whenever you see red things, they fire together. But then whenever you see circular things, other groups of neurons fire together. And then when you see an apple, all these groups are activated synchronously together. And then your perception of the world makes you do oh, it's an apple. So this is the kind of question we can go. Are you okay with that? So far, so good? So we can think, can we build a machine or how the brain classifies? And depending on the question, we can go both sides in this idea of more biological neural networks. Considering that we are going to stay most of the part of the presentation in these more biological neuronal networks, I have to sit. Sorry. One kind of big questions that we can go in these biological neural networks is the functional versus structural connectivity problem. So how we have the brain with a kind of fixed anatomical structure. So we have the neurons and the synapses. They can change with plasticity, but more or less they are fixed, or they change very slowly. But we can do very different tasks very fast. So I can talk to you, but if there is a fire alarm, I can run or not. Please help me if this happens. And then we can remember the supermarket list, and then we can recognize someone that's sitting there. So we are very good to do a lot of tasks. So the activity of our brain is changing. So if we measure this activity and define how this information is flowing, we are going to have functional network. So we are going to have a network of what region is correlated to other one, what is the flux of information from visual areas to more complex cortical areas to let me know that I have to run if I see something. And so besides the question of we can perform some kind of task, we can change the tasks very fast, more fast, and we would like to make very different functional connectivity patterns in a same structural connectivity or in a structural connectivity that change slow. So this is also a big question that can be answered or can be studied in the light of biological neural networks. So we can go to really try to reproduce the best of you can of biological neural networks that can do with same structural connectivity, very different functional connectivity. So we do not change the structural all the time, but we can have very different tasks, and our model should be able to do very different tasks. So with this in mind, we can go in a cycle that we do measures, like we determine functional connectivity using brain data, using brain signals, so electrical signals from electroencephalogram, magnetoencephalogram, different techniques. And then we have temporal time series that are not common when we are talking just about a neuronal network, artificial neural networks. So we have this time series, and then we would like to propose biological neural networks that have same kind of electrical activity of time series here. So we develop models, we do simulations, we develop models, and then we compare with experimental data, and then we try to improve our models. So I hope it's clear that it's different from when you are using a neural network? Okay. When we are using an artificial neural network to classify the different drugs that would like, or to classify different amino acids, we are not trying to improve how similar our network is with the network that we are studying. Like here, I'm studying the brain, and I'm trying to improve my network to make it similar to the brain. But when you are just using artificial neural networks to solve a problem, you don't care that it look like, that it perform the task the same way you do. You just want it to be good, to be performed with high rate. And here, we are more interested in really reproduce what is the network doing. So we can go to this structural connectivity problem, really analyzing how the different regions of the brain are connected, really where is fiber that take one region to the other, and the information can flow from one region to the other. Then we can build a matrix, and then we can propose which is the structural network of the brain. So for a human brain, this is very hard. We have 10 to 11 neurons. But for some animals, like C. elegans, for example, we have hundreds of neurons. We can really trace each neuron, and we can say which neuron is connected to each one. We can really reproduce the structural connectivity of our network. So when we don't know the structural connectivity, we try to infer, we try to compare to networks that we have, or that math had already studied. And then we can compare these structural brain networks with functional brain networks. So 1 point before, these networks, each node here and each link here could be different things, but let's think in the beginning at least each node is a neuron and each link is a synapse. So far we can think about that, and all the idea here is that I will tell a lot of ways to describe the nodes, the neurons, the dynamic of the nodes, then the synapses, and then the structure, the topology of the network. But we are really interested in understand anatomically, so this neuron has a synapse to this one, but has not synapse to this one, or this brain region is connected to this other brain region, and not otherwise. But we can also look to this functional connectivity, so we can also look to the time series. So when we recognize an apple, different things are going on in different brain regions, so we can also build a matrix of which regions are connected functionally, but not necessarily there is an anatomical connection, but their activity are correlated, for example. So if activity of different brain regions are correlated, we can say that they are somehow connected, and we can build a similarity matrix saying that visual areas and prefrontal areas are connected in our networks. All these neurons fires and these neurons fires together, so probably they are wired together. I don't know if there is a synapse there, but every time I see this neuron firing, I see this other neuron firing, so I will say that there is a functional connectivity connecting them. Questions? Are you with me at least? It's a walk, okay. So to do that, we can do in different scales. So that's the point. We can consider the brain and brain regions, or we can go to cortical areas like millimeters, or we can really go to neurons and ion channels, and single neurons here, or membrane potential and lipidic membrane here, and different models will be developed for different spatial scales, and different spatial scales will give different time scales also. So this is something that we have to have in mind. So if we consider this scale here, cognition and the nervous systems, this kind of problem, this kind of task would be here in 1 meter, 2. If we go to the scale of the brain, we are something here in our, if it's human brain, so we can have small brains for different animals. We can go and study just specific regions of the brain, cortex, thalamus, hippocampus, depending on the kind of question you are interested to. So we can go to the cellular level, or you can go to the molecular level and neurotransmitters and biochemistry and DNA and delivery drugs. And so you have to do many decisions in how you're going to study, how you're going to approach your scientific questions, and then scales are important for us to decide how deep is our model and the details. And it's always, you need always a good compromise. Like you cannot go to all the details, otherwise it would be a huge problem you cannot solve, but you cannot simplify it so much. So we have around 10 to 11 neurons, and each one do like 10 to 4 connections, so it's a huge amount. I cannot simulate that in my computer, but we do the best we can. So we can also look to multiple scales depending on the experiment we can do. So if we are doing like EEG, fMRI, EEG would measure electrical changes in milliseconds and seconds. If we go to fMRI, we have something like 10 seconds, and then it's more like blood. But if we have more blood in a region, probably have more activity, electrical activity, so it's different time scales, but they could be related, electrical activity and signal, both signal. So also the scales are different. Here we are measuring the brain, but we can go and measure like optical imaging nowadays. You can go and measure few neurons, like in rat brains, typically. So again, you can, depending on what you are trying to understand and model and measure, you can go molecules, synapses, neurons, circuits, networks, topology, or system, the whole brain, and cognition. So everything is very different and depends on different things that you are looking, like temporal, spatial, and topological. You can thinking of how the neurons are connected locally, how the neurons are connected globally in the brain. So it's a modular structure, it's a hierarchical structure. So neurons, close neighbors, neurons are randomly connected, but if you look to a larger scale, you're gonna see like a different topology, that visual areas is sending a lot of links. It's a big hub. And so if you look to space, you start with cellular level, so neurons, but also neurons have axons that can go like 1 meter, depending on the thing you are interested in. Like if you are interested in something like motor, there is 1 meter going until your foot for you to play soccer. And then perhaps for this kind of thing, if you are interested in how the signal goes so far, you cannot consider the neuron just a point, just a node, but you can have to consider all the details of this spatial neuron. But then we can only think not because the axon is so far, but because there are many neurons in 1 millimeter of the cortex, so you can stay in a different portion of the cortex, so you can thinking about populations of neurons together, more or less they do something similar, so you can go to brain areas, and similar idea with temporal things. So I hope This makes the point in order to talk about biological neural networks. We can mainly think in these 3 H's here. So this is from hockey courses, the previous courses that I've attended in this subject. So we can decide which neuronal model we are going to use. Then we can decide which synaptic model we are going to use. Then decide the network topology. Then we will have our biological neuronal network. Okay? So I will spend sometimes here showing a lot of different neuronal models, and then synaptic models, and then network topologies. And I hope you enjoy. So neurons. Disappeared my figure, but this is the important thing. So the brain is made of thousands, 10 to 11 neurons, lots of neurons, a number that it's out of our imagination. But until end of 80s here, there was no clear answer if it was really neurons, separated neurons, or if it was a tissue of activity. So we call neuronal doctrine, and described by Ramon y Cajal, which was the draws. But he really was able to really do sketches of what was going on with these cells. And then nowadays, this is what have drawn in the beginning of the Introduction to Machine Learning course. And this is the basic idea of a neuron. There is a soma, this main body of the neuron. And then we have these dendrites, and then we have this axon. So the information is going from the dendrites to the soma until the axon. So there is electrical activity passing to other neurons through synapses. So in our perceptron, this is just a circle and a number. And here we can go to how many details you want or you need, or depending on the kind of question. And this is what I will try to describe in the next slides. So the dendrites receives and process inputs. So other neurons, presynaptic neurons would be here. They send information that for us will be electrical current that enter in the neuron and pass here and then go until the axon to other neurons. Of course, this is already a simplification. We can have synapses all the way around, but to be at least with what is more common, this would be the idea. And so we can also try to model this spatial scale of the neuron, or we can think that the neuron is just a sphere, like the node, but there is dynamics. So the neuron has electrical activity. I think the numbers are huge. So in 1 millimeter, we have 10 to 4 neurons and 3 kilometers of wires. So these big axons. And it's hard for us even to understand cognitively what does it mean. But we can try to simulate thousands of neurons. And if you ask for biologists, there is a lot of more complicated things. Neurons are different in forms, in function, in different regions, in different species, animals. So they are doing different things, but we are going to use this exemplar model here and try to think that at least there are many things that they do similar. So let's stick with actual potentials for those who are used to, but I will arrive to that. So instead, it's hard to be sit down. Let's go. Instead of consider just the perceptron or few of them, I don't know, which would be a neuron in an artificial neural network that will be a number. I think it was like that, something like that. It depends on the function that it receives. We are going to consider a sphere. and not a beautiful thing like that. But this sphere has a dynamic. So this will have an activity, potential in time, that's a potential that is changing in time, a difference of potential between different parts of the cell. So this is a very typical behavior of neurons. So almost all these different zoo of neurons that I've showed before, they do actual potentials. What does it mean? So if you record the membrane potential inside the neuron and compare the electrical activity inside, they have this very characteristic form here. So the potential inside the neuron and outside the neuron, the potential difference between the intracellular and the extracellular medium has some rest activity or sub-threshold activity, I would say. And at some point, if it receives signal, external signal, this potential difference starts to increase. And then if it reaches some threshold, it goes all the way up until here and then decays in something in the order of 1 millisecond and half millisecond, and then it stay in a refractory period, and then recover and come back to the initial condition, okay? So this is the typical spike or fire. When I say that a neuron is firing, a neuron is doing that. Perhaps here in the perceptron, we would say that when it's here, it's 1, and when it's here, whatever, it's 0. So 0, 0, 0, 1, and then 0, 0, 0, 0. But depending on the kind of question, you really want to look for this dynamical activity, and is okay, the neuron fired in a certain point of time, and then we'll fire again. So we'll have a firing rate. So it's firing, or just go to 1 and stay in 1 forever. So we can study this kind of thing. So let me see. These refractory periods can be like it's a time that no matter how strong is your input, like if you give a lot of injection of current in the neuron, it will not spike again until it reaches some potential, rest potential here, and can fire again. So this is important for neurons. And this threshold that I mentioned that is not necessarily here is related to thresholds that we put in our sign functions in artificial neural networks. So some ingredients are related, and some ingredients can be important for the artificial neural network. So specific neurons have specific shapes, so you can identify when you measure all these small difference in one actual potential is from one neuron and from another neuron. It propagates along all this way without changing the form. So these actual potentials, if you measure here in the soma, or if you measure in a different region, you will see the same profile. But the sub-threshold voltage, so the activity that is happening that are not related to a spike, they decay. I have another slide. So this is more or less what's going on. If the neuron is receiving external inputs here and at some point, so there is an excitatory postsynaptic potential, so you do an injection of simulating another neuron here, and so the potential increase a little bit, then start to decay very fast, and then another stimulus arrive, and then it increase a little bit, start to decrease, another stimulus arrive, until a point that if you reach the threshold, so you have this all go up, and a spike emission, and a fire or a spike of the neuron, and then it goes back until the refractory region here, and then if you do a stimulus here, nothing happened, and then it starts again. So this is how to describe the actual potential. So, sounds okay? Little bit different from a number that receives input and you sum these inputs. But with this inspiration, sometimes you want a threshold. Sometimes you want to really look for that, just like this is activity and all the rest of the time you are not active. And external stimulus would be related to synapses that are arriving in a biological neuron or in a network. But in our network, this is coming like one layer and then after layer, then after layer. Here, no, we have this time, discrete time, and then we can treat as dynamical systems. So first verification of an actual potential was done in 39. Then they received the Nobel in 52, but in 39 they Hodgkin-Huxley recorded this actual potential from a real neuron, from a giant neuron of a squid. Okay, it's not a giant squid, it's a giant neuron. This is a joke in neuroscience, but thanks that it's a common squid, but with a very big neuron. So they were able to measure this difference. So what they did, they realized that neuron exhibits electrical activity. These ions exchange. There is ions changing from the inside of the cell and the external environment. So there is a variation in the potential difference between the internal and external environments due to this ions movement. So Hodgkin-Huxley experimentally measured this variation, and they proposed a mathematical model to describe this variation. So here we can go slow if you are not used to electrical circuit, but I think it's nice if you never see. Who have seen this before? Yeah, okay. Nice. So what they did, they measured, so they recorded, they did the experiment, and they said, okay, can we find an equation that reproduces this behavior? So they look to biological ingredients, and they describe each part of the neuron as a part of electric circuit. So they realized that the membrane, the cell membrane, could act like a capacitor, because we would have negative charge in one side and positive charge in another side. So they said, oh, maybe we can represent the membrane potential of the neuron as a capacitor. And then they realized that the ionic channels of different ions would be related to batteries and conductance here, so it would be easier for sodium to go from extracellular to intracellular in certain conditions, and for the potassium to go in the other direction. So they try to model what was going on with the circuits and the equations. I think I have everything here. So they said, okay, they did different measures of sodium, potassium, and leak currents, different ions that are just not that important in the time. And they say, okay, we can represent the membrane potential, the chain of ions of the neurons with this circuit. So a capacitor for the membrane potential, and this resistance and battery for sodium and potassium. So perhaps you remember, perhaps not, but if we define like the potential inside the cell and outside the cell, so if you look just to the difference, we can say, okay, there is a difference of potential here from, here is inside and outside, and here is outside and inside, so don't worry. But we know from physics 3, I don't know, that if we have a capacitor, the potential is proportional to the amount of charge that we gonna have accumulated in the plates of the charge divided by the capacitance. So if these charges are changing in time, but the capacitance is a fixed parameter that depends only of the membrane, this potential can vary. So if we look to variations in the potential and variations in the charge, we can rewrite that as variation of potential equal to variation of charge divided by the capacitance. So if they are changing in time, we can say that variation of potential in time is related to variation of charge in time, which is current, okay? So if charge is changing in time, we have a flux of current. The ions are entering or leaving the cell, so we can define a current here. And the current is related to the variation of the potential in time. So we can describe for this part of the membrane, not considering the ions that are moving, just the fact that the membrane capacity accumulates charge, we can say that the capacitance of the membrane times the variation of the potential in time, so how this potential changes in time, is related to all the current that is arriving in this neuron, okay? So if I do a stimulation in the cell, or if there is another neuron that is stimulating, or if there are sodium and potassium entering and going out, leaving the cell, we can add everybody here. Okay? So we have this relation of how the potential change in time is related to the amount of current that arrive. So the activity of this neuron in time depends on the previous neurons that are sending current to it. Okay? So the state of this neuron, the potential of this neuron is related to what is coming, but in time. And what is coming in time can be changing, and we can stimulate in different points of time. And when sodium and potassium are going inside and outside the cell, we have the other part of the circuit. So we can perhaps remember that when we have a resistor or a resistance would have is related to a conductance. So the resistance of a resistor is 1 over the conductance. So how difficult it is to pass is related to the opposite of how easy it is to pass. Okay. So typically in physics we use resistance, but in neurons we use conductance. So it is easy for the sodium to go to this direction or not. So the conductance is increasing, more sodium is passing. And then the potential difference in this resistor is related to the resistance and the current. So the current that is passing in this branch of the circuit is related to the conductance and the potential. So if we increase the potential here, more current is passing. If we increase the conductance here, more current is passing, okay? So for each for each ion, we can look how this behavior biologically. So this is what Hodgkin-Huxley did. So they looked to sodium, and they said, oh, okay, there is these ion channels of sodium. It behaves like this resistance and battery. So it's here in our circuit. So the current, sodium current, is the conductance of the sodium through these channels, and the potential difference here. So they said, okay, the potential difference here depends on the potential of the resting situation for sodium, so we can use Nernst potential to define this value, but I mean, it's just saying that there is a potential difference for the sodium from inside and outside the cell, and there is a conductance for sodium in this part of the membrane that depends more or less on how many ion channels we have, how many channels are open, And then there is a dynamical process here because the conductance is not fixed, so sometimes more ion channels will open, sometimes they will be closed, but then we can describe in this simple fashion, like G times V, the sodium and the potassium and all the other ions that are interesting for us or other kind of current that is passing along the neuron. Is it okay? I lost everybody? Okay. So we can do the same for the potassium. And specifically for this model, what is going on is this dynamics that is multiplying the conductance. And then I'll try to give an idea of what is this. But then one neuron can be described by the variation of the potential in time as a sum of all the currents that arrive in this neuron. And each current can be described by a conductance times the potential difference. So besides that, the conductance of each ion can have a dynamical behavior. So this conductance is not fixed, but it can work like channels that open and close in a first order kinetic dynamical equation. So if we have a fraction of the sodium channels that are open with a rate, a beta rate to close. So if they are open, there is a chance that they close. And if they are closed, there is a chance that they open. So every step, the channels can open. This is a first order dynamics that can be described by these differential equations, n here and x here, sorry for that. But the ways that these these activation variables change in time is given by a first order kinetic equation. So we have the probability of the channel to close and the probability of the channel to open. So which were closed in the past will be open with a rate alpha. So I hope this is okay, but if it's not, don't worry. Each Huxley realized that each conductance here was related to open channels, activation variables that could be described by this kind of differential equation. So for describe the behavior of the neuron, we need this first equation for the potential, which is the variation of the potential in time is related to the sum of all the currents, but we also need the dynamics of each ionic channel, and this is related to how many ionic channels of this type of ion was open. And at the time, they realized that these numbers, like the proportion of open channels for sodium, you will need M to 3 and H to 1, and for the potassium, N to 4. So like they did it experimentally. They determined this just to really fit the curves that they have, but they were able to do this with very detailed and biological parameters, so they really describe this behavior, and they were able to reproduce data. So with these equations in mind, they look to how these channels, ionic channels of potassium was open, this sodium, how they were changing, and then they decide these small numbers here, but in the end, they need 4 differential equations. So one for V, one for M, H, and N, and with 4 differential equations, they were able to reproduce the behavior of the neuron, so V in time can do an actual potential that was very similar to the real one that they were studying. So they have this part, that's the sodium's channels open, and then the sodium's channels start to close, so the actual potentials come here. The channels, the potassium starts to open, and they have the refractory period, so a lot of complications to say that, okay, with these 4 equations, that each term is biological, measured, and related to real things in the world, they could reproduce with the model, with these 4 equations, this shape of the actual potential, so they received the Nobel Prize. Great, everybody is happy. So that's the idea of doing using the scientific method to develop and test a model. So just to be everybody together, they realize the neuron exhibits electrical activity, so the neuron changes ions with the external environment. So there is a variation of the potential in time between internal and external environment, so we can measure that. So Hodgkin-Huxley measured that. They saw it was an actual potential, so they developed a mathematical equation to describe this variation. So they developed a model. The model was based in electrical circuits and differential equations, but could be whatever kind of equation. They described the same variation of potential in time. So nowadays, with this in mind, with a model in mind, we can go perform computational simulations to reproduce this variation of the potential and make new predictions. So if we cut the sodium of the medium, what's going on with the equations? How these equations change if we put more sodium, or if we change the conductance of sodium? So we can do predictions with the model, test, and then we can go to experiment and test again if our prediction makes sense. So we validate the model. So Hodgkin-Huxley model have been validated in many times. There is a range of validation, of course. It's specific for that kind of neuron, but can reproduce more or less different neurons for different animals, not just the squid. But I think this is also like to think about how you can do science in a specific example. So we can come back again. I don't know, what time should we stop? Fernando is gone. Maybe it's a good time, but I can. Yes, that's. So I don't know. Let me just to give the flavor of what's going on. Okay, with the idea that we received the Nobel Prize here, okay, now we have a model that has these 4 differential equations for a very real and biological inspired neuron. We know how to reproduce that. If we look to our artificial neural networks and biological neural networks, is it enough to know how the neurons work to build a machine that is similar to the brain? No, not yet. But then what can we do? Then we can think about different models. We can think about modifications of these Hodgkin-Huxley models. If you are interested in more biological ingredients, we can go from one direction. If you are interested in more simplified models, like a small dimension, we would like to do simulations of a lot of neurons, and we need better performance, so we can go in this other direction and try to simplify what's going on. So this is what I can do in the next slides. I don't know if we should take a break now, and so it's questions, not the time to breathe and start again in a while, but you can ask if you want. No? Yes, let's wait for after break. Okay? This is important. Please